Hello everyone. My name is Kunal Mitra. I'm a professor in Biomedical Engineering at Ford Institute of Technology, and I'll be doing today a webinar on surviving spaceflight to Mars, astronaut health challenges. As you could see that I'll be talking about two topics, non-invasive monitoring of cerebral blood flow and neurophysiological dynamics for spaceflight, and characterization of vascular tissue functionality during spaceflight using 3D bioprinted tissue. Along with me, I have today Mrs. Likhita Somasekar. She is a PhD student in biomedical engineering, and her research is focused more on the second topic, which is the using 3D bioprinted tissue to characterize the vascular tissue functionality. So let me first talk about this topic of non-invasive monitoring of cerebral blood flow and neurophysiological dynamics for spaceflight. So why this topic is important? I know this is a busy chart, but actually this comes from NASA. As you see, we are in space domain. So for us, NASA is the key to recognize what NASA is interested in. And if you look carefully, you're going to see that the red area is the area where NASA wants to really invest for the next few years. And FY 15-16 of the fiscal year. So right now you could see that we are in fiscal year 18, and then 19, 20, 21, 22, all are red. For the topics I'll be talking about: spaceflight radiation exposure, and the fourth one, which is the vision impairment for intracranial pressure. So these are the two topics which is of significant interest to NASA because of the long-term risk associated with the spaceflight. So what NASA does when they call out research proposals, they like to identify what are the challenges, and they will actually, you could see from the link actually from the NASA's website, they have like risk, gaps, and task. And if you could see there that the gaps task is sort of highlighted under gaps, this vision impairment is, they call it as VIP, and there they have this task which is of need, they need to be addressed, that they need to develop a tool, diagnostic tool, to measure and monitor changes in intracranial pressure, ocular structure, and ocular function. This is a very important topic for NASA, and I will not read to the whole details, but if you could see, this is from the cut and paste from NASA's website, that they have certain tools already in place, like a tonometer, fundoscope, and OCT, optical coherence tomography. They have been using this tools, but still the results are not conclusive. They are still looking for new ideas, and that sort of motivated us to pursue this research. So why we need to address this challenge? Because there is a term called space headache, and people use what a pain it is. So when you're exposed to microgravity, as in a space, your CO2 level, the carbon dioxide level, is much higher than what you experience in Earth. And scientists believe that this excess CO2 level in the microgravity environment in space is the reason for space headache. So there is a need to analyze that how does your cerebral functions get affected due to CO2 exposure. And again, another perspective, that in microgravity, your body, you may already recognize that fact, that when people go to space, there's a shift in body fluids, and your upper part of the body fluids tend to go more to your upper part of the body, and which increases your intracranial pressure, and this causes headaches. And so there is a need to see that how we can sort of measure that extra intracranial pressure, because we need to have a tool to alleviate the pressure once we know it is high, so that astronauts are not at risk. And the figure bottom sort of will tell you what are the challenges happens when you're in microgravity. You have changes in the visual activity, there's an intracranial pressure issue, then interocular pressure, they're all related, as you know. So these are and then the cerebral spinal fluid flow. So all these things are affected in space domain, and when you go in the microgravity environment. And with all the long-term Mars mission coming up, think of it, you will be about 3 years total combined, including your round trip in space. So if we don't address these issues of health issues, we are really putting the risk. And we're going to see also that along with the physiological changes, there is some mental issues which are sort of correlated, psychological effects. So we really need to address this physiological effects of intracranial pressure. So this is one what NASA has. There is another term for the interocular pressure. They call it neuroocular syndrome. It's NASA has been using different acronyms. The NASA is actually, if you in this domain, very well known for using acronyms. Another acronym is known as SANS, which is the neuroocular syndrome. So they have come up with all these articles. You could see what can be the issues with the space travel. Their eyes are at risk. Their vision problems. So all this has to be addressed because intracranial pressure and the vision, interocular pressure, they're related, and we need to sort of able to address that for a successful long-term space mission. So what happens in microgravity, as I already mentioned, that you have an increased CO2 level, and so there is a change in your blood flow, in your fluid flow in your body. So if you could see the figure on the right, that sort of shows you what happens to your blood flow in your body. So there's an increased fluid flow, which causes your pressure, your increased flow towards the top of your body, and that means your body has to react to this. You have an enlarged heart because of that. And as you know that we always respond to the changes, right? So your increased heart will result in, we want to eliminate fluids from our body. And by doing so, we actually cause challenges because your plasma volume and the efficacy of the orthostatic reflexes, which regulate your blood pressure, is going to decrease. So, and a result of that, during re-entry, your blood volume is insufficient to maintain the blood flow in orthostatic position. So that's the challenge we face. So we have to be very, very, careful in addressing these challenges. So what happens, again, when your cerebral blood flow is affected? So there are actually two mechanisms which comes into picture. The two acronyms are CVR and CAR, and I'm going to sort of expand that. CVR is cerebrovascular reactivity, which is the response in changes to partial pressure of your arterial CO2 levels, which is needed to maintain the central pH in your brain. And the other is the cerebral autoco-regulation mechanisms, which basically counters the blood pressure changes to maintain the cerebral blood flow to the capillary beds of the brain. So again, the excess of microgravity environment will induce this higher intracranial pressure, because that will result in also intracranial hypertension also, which is due to the shift in the body fluid. So our main question is that, as you know, that our body responds to the changes due to the autoregulatory mechanism. So the question is that, do our body respond to these autoregulatory mechanisms? Do they remain intact during long space flight? And is our cerebrovascular reactivity response changes during long-term space mission? So the impact I already mentioned due to this long-term space flight: increased ICP, increased intracochlear pressure, and increased cerebrospinal fluid production. So all these are sort of the result of the long-term space flight issues. And it comes along with headaches. We also talked about it due to increased CO2 level, maybe dizziness, nausea. And as I mentioned in the beginning, that this actually affects your mental health too, psychological issues. So we need to be really very careful to analyze these effects. So what is CBS dysregulation? So because why it is important? Because this can be actually used as a biomarker for many other physiological conditions such as TBI, Alzheimer's disease, which is very important research area right now. So we are even not focusing on space. You can actually correlate this research to other applications because they sort of have the same physiological representation. Like traumatic brain injury is a major impact, especially if you're in the sports arena. NFL, they invest a lot on concussion studies, and actually this can be related to concussion studies too. So we need to really understand the cerebral blood flow dysregulation mechanisms. So what happens? So CVR, so when you have a dysregulation, what are the effects? It affects your cerebral blood flow. And the CO2, as I mentioned, is actually a powerful vasodilator. So if an increased partial pressure of arterial CO2, it will cause vasodilation and it will decrease your PCO2 because that will result in vasoconstriction. And you want to maintain your vessels dilated, otherwise you're going to have an increased intracranial pressure, which will affect your even your vision in the long term. So as you have seen in the slide when I shown in the beginning, that NASA is looking for techniques to measure this intracranial pressure. One of the techniques is the invasive measure, which is a lumbar puncture, which is an invasive procedure, and it will need actually a skilled worker. So it is not easy to use. So we are in need of non-invasive technology. And these are some of the techniques which has been used by NASA. And of all of them, I'm going to talk a little bit about the first one, the ocular sonography, because some of the other result ones has not given significant important results. And MRI is a good tool, but MRI is not feasible for space, as you know that you cannot take an MRI machine in space. So based on that, the technique which has been most commonly used so far is ocular sonography. So basically what they measure there is the optical nerve sheath diameter, because that can be related to your intracranial pressure. Generally, it has been accepted if your optical nerve sheath diameter is greater than 5 millimeter, your intracranial pressure is going to increase by more than 20 centimeter of water, and that means you have hypertension effects. So still, this technique is not a reliable technique because no reliable interlinear relationship has been sort of demonstrated to show that optic nerve sheath nerve diameter can be related to ICP very explicitly to hypertension and ocular intraocular pressure. So what we have been working on for the past few years is on near infrared spectroscopy or NIRS. Why? Because it is a safe method, which is non-ionizing method, and it can be used to image monitors to monitor humans and animals. So it basically is a light technology. You have a source and light detectors which is placed at a certain distance, and you actually measure the signals from there. And the distance, between the source and the light is important to characterize the signal. So I'll talk about how the NIRS works. So you put a source light to the fibers and you collect the lights, and based on the signal what you detect, you can take information about your intracranial pressure and what your brain activity sort of in a way. So it's again based on light technology, and it uses wavelength of light in the range of 700 to 900 nanometer, because that's sort of the best range for light to propagation to measure functions, physiological functions through tissues. And what basically NIRS does is basically measures the concentration of different species, the most important being oxygenated and deoxygenated hemoglobin in the volume you want to measure. So that's sort of the parameters you look at. So what really NIRS measure at the end as an end user is what is known as regional cerebral oxygen saturation, which is RSO2. So if you can measure that, which is again based on, as I mentioned, on oxygenated and deoxygenated hemoglobin, and we can sort of correlate that function to your brain activity. So what we'll be really measuring is RSO2, which is the regional oxygen saturation values. So how do you sort of, able to do that first in a lab setting? Because we need to do a lot of, when NASA really wants to do space research, the first step is ground research. So how do you sort of, do that? One way to do that effect is you induce hypercapnia. What is hypercapnia? It's basically nothing but increased CO2 level in your body. So if you can increase the CO2 level, then that's going to increase your POCO2 level, and that will result in your vasodilation and increasing your RSO2 level. So we can then correlate these functions to the RSO2 level. So partial pressure of CO2 is a critical parameter which you want to measure, because then I can correlate these measurements to the regional oxygen saturation values, which is the RSO2 value, which is a NIRS will measure for us. So why this is important? Because if there's a dysregulation of your body function on the cerebral blood flow, then the response of the regional oxygen saturation, the RSO2, will be different due to hypercapnia. Again, what is hypercapnia? As I mentioned, we induce excess CO2. So if there's an excess CO2, and if your body is not able to maintain the regulation, you're going to see dysregulation, and you want to characterize that. So, but before that, what you need to do is to collect your baseline, because my regional oxygen saturation baseline will be different than you or anybody else. So our first goal is that when you do this research, that's why when you want to send the astronauts to space, you want to measure the baseline before they go to space long-term mission, because from then you can monitor that increase of the RSO2 levels from the future from the baseline values. So our goal is to first maintain this baseline values, and then you can, from there we can see that if they will return to the baseline level after the stimulus. If they don't return to the baseline level due to the stimulus, that means you know that they have a dysregulation, because it is not a regulatory. If the regulation is maintained, it will come back to the baseline values. But if it is not, then you can tell that they have a dysregulated, the cerebral blood flow dysregulation has occurred. So, as I already mentioned, that MRI is a great tool, but it is cost prohibitive to use it in space, and it is not portable. So there are some other techniques which has been also used by NASA, which is called transcranial Doppler measurement, TCD. The challenge is that the TCD really doesn't measure the cerebral blood flow directly. It sort of measures the cerebral blood velocity, which is really not a true indicator of the cerebral blood flow. So we are still in a need to develop inexpensive, which is a key word, non-invasive, which is a key word, which can measure the intracranial pressure and the cerebral blood flow. So just to give you a perspective, like when you do exercise, if your metabolic rate, when you are, so your blood flow is also going to first start increasing because of your metabolic activity. And when you are doing activity, your cerebral blood flow is going to keep on increasing up to a certain point. When you do a heavy exercise, actually you see a dysregulation, and that's the reason you could see actually your cerebral blood flow starts actually decreasing due to heavy exercise, because your body is not able to respond to the cerebrovascular changes. So that's why again this is a relative term. The mild, moderate, heavy exercise is based on your baseline value. So that's the reason it's very important that we characterize the baseline value, and from there we can get a perspective that if you are doing maybe heavy exercise or any other conditions that can affect your cerebrovascular response. So it has been well documented that cerebrovascular response will increase during moderate exercise, and it will decrease during sleep. And it also actually affects your circadian rhythm too. And as I also mentioned, that even you are focusing on space research, this has got a major application in traumatic brain injury, which is actually a major sort of research area when you're suffering concussion, for example, or any other head injury. So we can actually use the same sort of algorithm correlation to characterize concussion studies as we are doing right now for the space-based application. So what are actually the open questions right now we need to understand? When is your autoregulatory mechanism compromised? Is it maybe after a few days in space, or if you stay in the space for 2 months or 60 days or for 90 days or up to 2 years? Who knows? So these are the open questions we really are able to understand the effects of long-term mission. And also is your response change due to CO2 exposure, because you're going to be exposed to more higher level of CO2 than here. So these are actually unknown. And as I mentioned, that every population is going to respond to these changes differently. So will the same effect or the same environment affect different individuals differently? So how do you sort of regulate that environment? And what is the best method to actually characterize the cerebral blood flow dysregulation? So these are the challenges we are actually going to address. So what we have done in the past couple of years has done a lab-based study as a starting point, where we actually developed a breathing circuit where you'll be breathing CO2 and try to see if your cerebrovascular regulatory responses are actually affected by that function, because you're breathing excess CO2. And so this was what we did actually in our research, that I'll show you a circuit in the next slide. We asked the individuals to inhale like air for a few minutes, and then we put them to the hypercapnia challenges, which is basically breathing a mixture of air and CO2 for a certain amount of time, duration of time, 5 to 10 minutes, and then we let them recover for a few minutes. And then we actually want to look at the response. We did the studies on about 26 healthy subjects. And the reason healthy subjects, ideally our long-term goal, actually our next goal is to actually do it on athletes who have suffered concussion. But even if before we do that, we actually first want to do on a healthy subjects so that we can characterize all the baseline values effectively before trying it out on concussed individual, because they don't have a baseline properly set. A healthy subject will have a much more stable baseline. So that's sort of our goal. So this is our study. You could see that we really instrumented the individual, and this is our main signal F. We used a non-equinox infrared, the near infrared spectroscopy system. So it's like a headband. You put the sensors here, and then we are monitoring all the physiological functions. We did an ECG because we want to make sure the patient is stable, and we have all other basic measurements, heart rate, all going on during the performance of this test. And you could see this is the breathing circuit, which is the H, and they're wearing a mask, and you may not be able to see the tank. See this pipe, it is connected to this tank, which is a mixture of air and CO2. And here is the valve. So initially, as soon as you want to close the valve, we are controlling the amount of air and the CO2 flow. So some basic information about this NERS system. Basically, NERS system will contain, the main thing is that how many wavelengths of light and how many source pair you're going to use will sort of dictate how a system is, effective or not. They initially started with only 1 and 1, like one sensor and one source, but now they actually want to put multiple sensors so that they can actually gather more information and improve. And the other important challenge is your data update rate. So a standard rate is actually 4 seconds, one update every 4 seconds, which is actually not great if you want to really look at some quick response. So the one we used, actually that has got about 18.75 updates per second, so it's actually a high resolution. Of course, as you know, with any high resolution, the cost. So we actually had these two systems. We tested one low-end signal system, which has an update of 1 update per 4 seconds, and then we have a higher refresh rate system, which was more expensive, but you want to compare the two functionality. So you could see here how the source and the detectors were placed here at a certain distance, and we are able to collect the signal. And our goal again was to measure the regional cerebral oxygen saturation values. So here are some of the results. So what we did, we asked the individuals how many minutes or hours to exercise per week, and based on that, we actually created three exercise factor levels. And I have actually the next slide actually like how many hours one means they exercise at a different rate. I'll show you right here. Like level 1 is no hours, 0 hours a week, and then level 2 is greater than 0 but less than 10, and level 3 is more than 10 hours a week. And so what we are able to do, we are able to measure the regional oxygen saturation values as a function of the entitled CO2, because that was sort of our goal we want to sort of measure. So by doing so, we could see that our response was very linear, and we got a strong correlation because these are healthy subjects. So if you increase their arterial CO2 levels, they should respond by higher level of regional oxygen saturation values. If they have a dysfunction, you actually should see a negative slope, right? Because they're not able to respond to the increase of the CO2 level. So these actually proves a very nice hypothesis that on a healthy subjects, your regulatory functions are maintained, because by increasing the CO2 level, they're able to respond by increase of regional oxygen saturation values. And by doing so, they're able to maintain the regulatory mechanisms, which is very important component for our study. So as I already mentioned, we have 3 groups, and you could see in a box plot sort of, you can just see how, if you're in healthy subjects, how their ROSO2 value is significantly higher compared to the person who doesn't exercise at all versus limited. So this sort of study was sort of a baseline that how important also is exercise can have on your regional oxygen saturation values, because actually that can actually have a major impact also for the astronauts too, because they may be able to do some controls by using certain amount of exercise time per day. They always do, but this will be very important for them too. So we also did some modeling too. But so in a conclusion, we can just say that we are able to successfully use the RSO2 value to correlate the cerebrovascular changes in response to CO2 inhalation in healthy subjects. So, so again, what are the current challenges? We have to really able to see that will the response be affected by the long time CO2 exposure, and how long they can sustain this response to abrupt changes in the blood pressure. So these are the CAR responses. So we need to be able to address these issues. So I forgot to mention, actually, I had two, as you could see, I had two presentations, and the total will run about 45 to 50 minutes. And I know that you're already typing the questions, and the presentation will be actually recorded for you at the end, and you can see that they can ask the questions in the pane on the right of the screen, or we can be answering them at the beginning. So how do I? This one. Oh, okay. Okay. Sorry about that. We have two present. The file size was too big to do it in one slide, so I sort of have it in two slides. So the next part is what we have been working on, using a unique 3D bioprinted tissue to characterize the vascular tissue functionality. So why this topic again is important? Because when you're going to, again, this comes from NASA, like the gaps, when you're in the space, this long-term space mission is actually going to affect your cardiovascular system because of different things, because of the microgravity effects as well as the radiation exposure. So we want to really look at this effect of the degenerative effects, especially our main goal here is on the cardiovascular tissues. So we want to look at the effect of cardiovascular tissues due to the effect of microgravity and space radiation. So some basic facts: when you're in space radiation, you have different types of energy, high energy protons and heavy energy ions, and they really will significantly deliver high energy radiation dose. And just for your information, just you can just see that a 253-day deep space mission is going to actually give you about the same amount of radiation as if you're taking a CT scan every 5 to 6 days. So that will give you a perspective. And Mars mission can easily go up to 3 years. So that will be, and this is 253 days, is less than a year. So be prepared to get exposed to a high dose of radiation when you're exposed to space missions, when you're going to space missions. So that's actually a very important reason that NASA is very interested, because this long-term radiation is actually going to affect your cardiovascular system. Of course, cancer is another thing, but that's beyond the scope of this presentation. Our research is focused on cardiovascular disease. So what is the challenge in space radiation? The functionality, the integrity of the endothelial cells and the smooth muscle cells, which are very key components to maintain the vascular functions, are affected. And actually there is very little information known about the effects of microgravity and the space radiation on the endothelial cells and the smooth muscle cells. So we really want to sort of analyze that effect. And in fact, there is some data recently done that some atomic bomb survivors, they have a elevated risk of cardiovascular endothelial dysfunction from a dose as low as 0.5 gray. So it's very important to analyze this long-term effect of space radiation. So we really want to do a very comprehensive study. So what way we can do? I mean, one way is definitely use an animal model, but that will need a lot of sacrifice of animals. So one of the unique thing what we have developed over the last couple of years is a 3D bioprinted tissue, which is a very unique topic by itself, but I'm going to go over a little bit in this presentation. But by using a 3D bioprinted tissue, we are able to do a lot of controlled experiments under different conditions, which will be not very possible if you're using animal tissue or any other model. And the 3D printed tissue is also very ideal because we can actually put any type of combination of cells, or so that you can change the characteristics of the tissue and you can print them and do all the functional and the physiological and the functional and the biochemical analysis we need to do to characterize it. So that's one of the advantage of using 3D bioprinted tissue to characterize the vascular response during space flight. So in a nutshell, what happens, how do your cardiovascular system get sort of affected? So the angiotensin 2 is the one which is sort of the unwanted phenomena which happens, manifestation happens in our body, especially more when it's exposed to microgravity. So you want to really decrease the effect of angiotensin 2, and because that will result in an increase of blood pressure. So one of the goals of our research is to see that how can you actually reduce the risk of increase of angiotensin 2, because by reducing the angiotensin 2 production, we can actually prevent the cardiovascular disease, because angiotensin 2 will result in vasoconstriction, and vasoconstriction will result in increase in blood pressure. So what is our goal? So what are the assays we are going to use to understand these cardiovascular dysfunction mechanisms? The important things we are looking at is nitric oxide levels and the oxidative stress, because nitric oxide should be abundantly produced by our body. And when you have a cardiovascular disease, actually your nitric oxide production is diminished. And we are going to be able to measure the nitric oxide level to see if your cardiovascular dysfunction has taken place or not. So as I mentioned, the decreased production of NO in many pathological conditions, especially in response to angiotensin 2, which I showed you upregulation in the previous slide, will cause serious problems in endothelial equilibrium and vascular functionality, which is a leading problem of CVD or cardiovascular disease. So you want to really block the angiotensin 2 receptor, because that will restore your vasoreactivity, and the NO expression will be sort of restored in this if you can block the angiotensin 2 receptor. So we have been working on 3D bioprinting tissues, and by bioprinting 3D tissues, we are able to actually construct large tissue constructs to be able to do this research. And what is the advantage of this 3D printed tissue? Again, we can put vessels of different sizes, and we can do a whole range of parametric study using a 3D bioprinted tissue to characterize the cardiovascular effects on the long-term space mission due to both the microgravity and the space radiation effects. So 3D bioprinted tissue sounds good, but actually pretty challenging, because as you know that we have a lot of extracellular matrix in our body, right? So that makes up our tissue. So we have to make them as like a recipe. So what you do, you take cells and we take different recipes so that you can simulate your extracellular matrix in our body, which is present. So the main goal in a 3D bioprinting is that what type of recipe do you want to put it in so that you can create your real 3D bioprinted tissue. So our goal again is actually in this particular presentation on cardiovascular disease for long-term space mission, but actually there's another important, few other important that you can actually use this for drug delivery applications. And the 3D bioprinted tissue actually have also major impact, as I said, on the cancer treatment, because when you do chemotherapy, what is chemotherapy? People will use different drugs and see how your body responds, which is again a challenging task. But if you have a 3D bioprinted tissue, you can run a whole bunch of assay and see what type of drug, for example, will work best for you. So you can actually do a customized chemotherapy treatment very easily, because what they can do, they can actually take the cells from your body, like the stem cells, and they can actually create a 3D bioprinted tissue which will emulate exactly your body tissue, and they can do all range of tests using chemotherapy drugs and tell you, okay, for you this dose is going to work best. So it is really a customized cancer, personalized medicine, which is another application we have in our mind to use for this 3D bioprinting. So in some other perspective, that it takes about 12 years and 1.8 billion to get a drug approved by FDA. Just think of the cost involved. And actually 90% of the drugs will actually not go through the human and the clinical trials. So this can have a significant impact, on the 3D bioprinted tissue if you can really successfully do it for drug development application. So that's actually another motivation in parallel, in addition to the cardiovascular disease, for space applications, which you're also working on. So we need to first understand what is vascular dysfunction. So your normal endothelial function is basically will maintain your contraction and relaxation mechanisms. When you have a dysfunction, you have an unbalance. That means you have what? More of vasoconstriction than vasodilation. So we need to be able to sort of overcome the vascular dysfunction in order to maintain your cardiovascular functionality or the vascular function. So why we are looking at nitric oxide? Because nitric oxide is a powerful indicator of maintaining of healthy cardiovascular functionality, because it dilates the blood vessels and it reduces the release of other superoxide radicals, which will sort of reduce your cardiovascular functionality. So you want to really maintain the nitric oxide level in your body. So again, nitric oxide and ROS sort of goes hand in hand, the reactive oxygen species. So our body, when you have imbalance, we actually produce a lot of these free radicals, so which you don't want. So we want to actually, you want to reduce the ROS generation and increase the nitric oxide level. So that will be the true improvement in your cardiovascular functionality when we are exposed to microgravity. You want to sort of improve those two effects. So as I mentioned, sort of show you in a long term our goal. So again, when you are exposed to microgravity, you have vascular alterations that causes oxidative stress that will reduce your nitric oxide level and increase your reactive oxygen species production and will cause cardiovascular disease. So our goal is to that see if you can develop some angiotensin II blocker, which will sort of help you to restore this nitric oxide level and prevent cardiovascular disease. So that's sort of our goal. So there are a lot of elements included in this research, I know, and one is really bioprinted tissue and then the nitric oxide level. But I'm trying to sort of give you a whole perspective how these two things can be sort of combined to address this very important research of cardiovascular disease for space applications. So again, the angiotensin II, as I told you, is which will sort of will reduce your nitric oxide level. So you need to sort of have a angiotensin II blocker, which will maintain your nitric oxide level. So I already mentioned our goal is to 3D vascularized tissue construct so that we can perform a whole bunch of parametric study to enhance the tissue functionality. And we are actually, I'll tell you some details of how you're doing the 3D bioprinting. We are actually using an extrusion-based system to bioprint a tissue, and we are doing a lot of characterization studies, biochemical as well as functional characterization, and we are actually monitoring the nitric oxide production level, which is because that's key to assess the vascular dysfunction. So these are the important parameters, broad objectives we are trying to achieve. And we have the fortunate advantage of working close to Kennedy Space Center. And before you go to ISS, International Space Station experiments, we will be able to do a lot of controlled experiments in these simulators. As you could see, these are some of the microgravity simulators which we have been using at Kennedy Space Center, which is like a half an hour, 40 minutes drive from Florida Institute of Technology, where you're able to do a lot of this important testing before we're able to put these experiments on ISS. Because NASA wants ground-based. But this is an extra step beyond ground-based because you're able to use these simulators present at Kennedy Space Center. So what we did actually, 3D bioprinting is an important aspect of our research and quite challenging. So what we actually did, we developed an in-house 3D self-contained bioprinting system. This is how it looks. Because a lot of times people will work on a bioprinter inside a safety biocabinet, but it may not be very easy to work on because, for example, if you're working on a drug development, you may be continuously able to have an access to your printed tissue so that you can put different chemotherapy drugs and test it. So we want to develop a tabletop system which has got it may be hard to see, this is a plexiglass chamber. So it has got a self-contained environment which is clean, and it has got the HEPA filter, so we can actually print inside this whole thing. As you could see here, these are the heads and this is your stage. So you're able to 3D bioprint right inside this chamber. So it is a very, very important advantage of a self-contained system which you can use compared to of a biosafety hood system where it will be much difficult to work on. So again, you know, for us to maintain the cells you want to maintain at 5% CO2 and 37 degrees, we are all able to maintain. And this is the laminar flow conditions. Hood, if you're familiar with a biosafety hood, it gives you a laminar flow. And this particular is system which you have developed sort of simulates the laminar flow conditions. And these are some of the designs of our pneumatic heads. The reason you have multiple heads because our goal is to print a multicellular components. So we can actually use each of these heads and mix them with different cells so that we can have a complex tissue of a multicellular tissue organ we want to print. So it has got the, right now using one or two, but you have the capability to use multicellular, all the heads on, at the same time to printing. So this is our extruded design of a 3D bioprinting system. And if you are familiar with liquid solid modeling, so you need to first have a solid modeling program. We are being using here PT Creo. Florida Tech has a university license to use that. You can use SolidWorks. So basically you create a STL file and then you slice it, and we write our own custom G code, which is then transported to MATLAB, and then we run the codes to print your structure. So you could just see here, this is one of our structures. How does it look like? And have some more pictures. We also actually been working on some models so that we can optimize our bioprinting parameters. So it's a huge challenge project, and I'm trying to sort of present to you maybe in 20 minutes the bioprinting along with the cardiovascular disease functionality. So just to give a perspective, you know, in a printer you need needles. If you just change the needle size, you can have different printing quality. So that itself can be just a few days of research to just see for one particular tissue what type of needle diameter is going to work best. Then what will be your printing temperature, substrate. So all this, will come into picture because the two things we're looking at will be the cell viability and the resolution, because these are the two things you want to be able to deliver when you're printing a 3D bioprinted tissue to compare with that of a native tissue. Because if your cells are not viable, the tissue is not going to survive. If the tissue is not very high resolution, you cannot do a lot of imaging studies. So these are the two parameters which you want to work on. So this is how it shows. Again, we are using some of these ECM materials, extracellular matrix of sodium alginate and gelatin. We mix them and then we print them on a petri dish, and this is how our bioprinting process goes. So bioink is the term which is most commonly used, so which is nothing but a recipe of your cells along with the extracellular matrix which makes up your tissues. And it is a highly challenging task because you have to have a right recipe of all these components, and you have to, once you mix it, that's not only the end of the job, you have to extrude it out. If you make it too viscous or if you make it too less viscous, it may not extrude properly and you may not be able to print properly. So these are a humongous amount of challenges involved in order to bioprint a tissue. And since you're looking at cardiovascular functionality, we want to use actually human endothelial cells because that sort of is the lining of your arteries and veins. So we have been using HUVEX right now and with different cell counts to perform this bioink formulation. So again, you could see some of our structures, how they look. We are using different combinations. Actually, we have changed, even as recently we are using, for the last couple of days, a different recipe to improve our 3D bioprinted tissue structures. And once you bioprint them, what is our goal? Like you think of your tissue, you have blood vessels in your body, right? So once you print this, we actually want to create a channel by sacrificial printing so that that can be your arteries and veins in your body. And you can supply nutrients to those channels so that a tissue can be actually kept alive. In fact, we are part of NASA actually have started what is known as a vascular tissue challenge. There are 7 teams in the nation and we are one of the 7 teams. They're competing. NASA wants to print a 1 centimeter. This is very thick actually. We are nowhere even close to that. Nobody actually. A 1 centimeter thick, which can be kept fresh for 30 days. And if you can do that, then NASA will give you a prize money. So everybody's trying. We have till 2018 end to sort of come up, and there are 3 prizes, 1, 2, and 3. And I know and NASA does a sort of an every January, like a meeting. Our next meeting is coming up. Nobody's even close to it. It's a very challenging task, but everybody's getting there to print that thick tissue. 1 centimeter is pretty thick to bioprint and able to maintain that functional characteristics. So when you go to space, as you know that this will be on a payload, right? So we actually have been working with a company, though they have developed this live cell imaging arrays. So once we bioprint them, this will be like a slots. And so we can do some live imaging of these tissues, of the bioprinted tissue when they're exposed to microgravity. So that's sort of our goal, to be able to use that for this research, because you need to real-time monitoring. And by partnering, they call it this, developing these biochips, we are able to monitor and perform all the assays we need to do. So you can do a lot of characterization, which is biochemical, and some are functional. And I'm going to go over some of them. Like we have to do some rheological studies to see the viscosity. Then you have to do live data to see how many cells, because it's a mechanical process. Think of it. You are mixing some uh, polymer materials with cells to create a living tissue. So you need to make sure that your cells are viable. And then we are doing histology to see, as you know, when you do for example, you have some skin lesion, you go to a dermatologist, right? They will take a tissue from you and say, hey, I'm going to send it for biopsy, and basically nothing but histology, and they will send you, okay, we'll get back to you in a week or so with the results. So we need to do a histology too to see if the tissue maintains the functionality. So we are doing all different sorts of characterization studies to see if this tissue is really, a true functionality of a native tissue which you'll see in your body. So this is some example of some viscosity of the shear stress, because very important for a bioprinted tissue to be able to maintain the viscosity, because as the shear stress increases, you want to decrease your viscosity, and it sort of follows the trend. So this is a live data assay, which is a very important component of this research, because you could see here that after bioprinting, this is the one which your control cells. Of course, everything is alive. But when you bioprint, think of it, the bioprinting is an extrusion mechanical process. You are going to lose cells, some dying. But the more you can keep it better, healthy, living cells, that means your tissue is much more close to a real native tissue. And you could see that this is after printing, we have a significant success rate of maintaining lot of live cells compared to the control. That means our bioprinting process is actually very successful. And this assay is known as live dead. They use a calcine, which is a green stain, and this EDM homodimer, which is a red stain. So that will give you a function of live is to dead function cells. So what is the challenge, as I told you, for NASA? They want to print and see if you can keep the cells alive for long period of time. So we did some studies up to 7-10 days and see if we are able to maintain. So you could see between day 1 and 2 and 3, we are actually able to maintain it pretty quite well. Days 1 means 1 day after bioprinting, we of course keep the tissues in an incubator maintained at the right conditions, 5% CO2 and 37 degrees C, which is a typical incubator temperature. And we have been very successful with our actually bioprinted tissues. So we did some MTT assays, which is again tells you information about the viability of the cells. But I want to show you some nitric oxide, because that's one of the important assay which you want to look at, because it's going to give you some functionality of your cardiovascular disease or not, because nitric oxide is the key biomarker we are actually using in this research. So what we did actually in our lab, we are going to use that as a marker before we go to do some microgravity environment. So we use some chemical to increase the nitric oxide level and to decrease it, and see if our system can recognize that. And we are able to very successfully able to do that. So we have a control, and if you increase some nitric oxide level, you could see the images sort of go brighter. That means nitric oxide level has gone up. And then LNAME is SNP, sodium nitroprusside. That means it increases the nitric oxide level, that chemical, the biochemical agent. And then the LNAME is a biochemical agent which reduces your nitric oxide level, because it will bring it down. And we are able to see this effect very, very confidently, I will say, so that we can say that our assay works well. So this is your control. If we increase your nitric oxide level, it goes up, and if you decrease it, it goes down. So we are confident in our assay before you want to go to the simulating test. And there are a lot of other tests we can do, the functional tests like myography, which will give you some information about the tissue force, like whether it can sustain the force functionality. So that one you can do. Histology is a very important, as I mentioned, like when you, again, like a biopsy, we do staining and we look at the cell structure. You can do immunochemistry, you can do real-time PCR. So for the purpose of time, I want to sort of really leave some time for question and answer, which is because that's will be actually intriguing part of our talk, that how what is the challenge you see while doing this research. So I just want to conclude that we were able to successfully print this 3D bioprinted tissue and able to maintain the functionality of these tissues for extended period of time. And our initial study sort of tells us that we're able to characterize the nitric oxide production level. So our next task is we are continuing the experiments with the microgravity simulator and going to the ISS for the future experiment. And thank you for your time, and I will go for questions. Okay. So I'm reading out the question. I'm a physician. My idea includes creating a blood laden using the the blood bladders using astronauts' blood and using it to increase the return to the heart during these effects. So yes, I mean, if you are able to bioprint, that's, I mean, we are open to printing any type of tissues and organs we like to work on. So if you have an interest, you can contact us. Right now, as I said, we are focusing on endothelial cells because we want to print the blood vessels and for a cardiovascular functionality. So we should be able to print any tissue combination. Our recipe may change because, as I said, we are mixing alginate and gelatin, and so for particular a type of other blood bladder, we may have to look a little bit recipe, because end of the day it's the recipe which matters, because that recipe is what is going to represent your native tissue. Yes, it is a very, you have our contact, we can interact and try to see what type of extracellular matrix materials will be able to work best for your type of application. Okay. Okay, sorry, we have your email and we'll email it to you, and you also have our email too in the beginning of the first slide. I will again come there if you, and my email is also there in the first slide here. So if you want to get our email, we'll be happy to sort of work with you and able to bioprint your tissue for your application. Okay. Any more question? Okay, I have another question. How does the bioprinted tissue work? If you can grow the tissue for experiments, can you not grow it in for replacement organs? Obviously. So that's actually, regenerative medicine is all about, growing organs. But as you know, that organs is a challenge. I mean, the whole, if you look at this, maybe this slide, that how do you want to develop, if you want to develop a very complex organ, how are you going to do a solid modeling of it? So that's actually another challenge of printing complex organ, because you're able to first create, for example, if you create a heart, it has got so many arteries and veins, vasculature. You need to create a solid 3D models first. So instead of doing organs first, you have to take a step back and do complex tissue and able to look at its functionality and then go to the organ level. I mean, yes, people have been working on ears, those are things, but complex organ is challenged because you need to first have a solid model of the organ you want, because that's the reason, as you could see, this is a very interdisciplinary field of biomedical engineering, working with mechanical engineers, because you need to know a good solid modeling background, able to do a bioprinted tissue. It's not like a pressing of a button. So this something is going to be very important for us to be able to analyze that. So we are taking a step at a time, tissues, and then heading towards the organ. So it will take you, if you go back to my presentation, I have it here. I have some quantitative information about the question is that how long does it take to accumulate 0.66 sieverts? Would it accumulate, would it affect the astronomers while on Mars more or less than in space? Yes, I mean, anything, as I said, I gave you some data here, I think. Yes, like if you're in the space for 253 days, which is not actually, Mars mission is going to be close to 3 years, you're going to actually, even the shortest, I think, if you can, there are some companies looking at like 1.5 year missions, because they're using like a trolley system to bring it back. So you're going to be still exposed to at least 0.66 sieverts, which is about 5 to 6 every day interval of a whole body CT scan. So yes, it will be a significant amount of radiation dose will be accumulated. That's the reason if you actually go back to the first slide which I have, that why NASA, this is actually NASA's #1, space radiation. I could see this is going to continue because Mars mission is not going to be, see, if you see all the reds, this is the NASA's biggest threat right now, space radiation. And that's the reason it goes beyond fiscal year 27 right now. So at least 10 years now they want to keep doing, because there'll be so many unknowns from the Mars mission that need to sort of address that. Have other question. Next question. What other effects of the space headaches have on the overall health of a human? As I mentioned, that is in a sense, this is the cerebrovascular effect. So you have two mechanisms that come into picture. As I told you in my presentation, the cerebrovascular, these are the two mechanisms, right? The cerebrovascular reactivity, which is the response of the arterial CO2, and the other is that the CAR, which is the response to the cerebral blood flow, and they are affected. And actually, as I mentioned many times, that is the physiological thing is actually related to mental. In fact, there are parallel groups of NASA right now who is actually looking at the psychological effects of long-term mission, because this is a very, very serious threat, because excess CO2 is going to affect the long-term memory. That's the reason I also, if you look at my slides, I have actually long-term goal is about Alzheimer's disease too, because long-term exposure to CO2 is going to affect many of these health conditions, and a lot of unknowns need to be identified for that. Yeah, do we have a future where you can 3D printing nerves and neurons? Yes, it is doable. Again, the resolution is going to be a challenge, because when you're printing, neurons and nerves, you're looking at very high resolution. So a lot of this is going to be printing. We are actually working on the sacrificial bioprinting method, which we believe will be the way to go for such application, because you can able to print sort of any size on your once you have a sacrificial bioprinting technique. So we strongly believe that the sacrificial bioprinting technique is going to let us able to print any type of size of neurons, because if your channels is made, all the sacrificial does, it extrude out the material you don't want, and the channels will be created. My next question, were there any long-term effects of the trials done on the test subjects? I'm assuming this is on the radiation, on the radiation aspect. Yes, there are some studies, I believe, has been done on the hind limb studies on the mouse. I'm trying to. There has been some NASA. There is some very limited work has been done by NASA on the long-term on the mouse studies. Hind limb, they do it on a hind limb, because basically they put in, that's a standard technique, and they put them in the space mission, and they have seen some nitric oxide as not be, that's as we are looking at, but there are some effects which has been shown on due to the hind leg studies done by NASA, that it does affect the cardiovascular system. So if you have, there's one more question, two more. Okay, what is with the physiological, psychological effects? Has there been any short-term effect caused by SANS, such as decision making? Yes. In fact, I'm not that, in that group myself, but actually at Florida Tech, we have our human factors group actually who is working, and our goal is to sort of work with them. So they're actually, in fact, NASA has their funding on the physiology part as well as the cycle. In fact, there are NASA call proposals. They're actually talking for both these applications. So I know that this is a work which I'm not working directly, but work is going on in this area. Yes. And the next question, is there cortisol effect in spaceflight because there is a connection with the renin-angiotensin system? I believe so, and so we are actually going to working through our research too, but I do believe, yes, there is an effect. So we are approaching the hour's time, which is what we have, but you have, thank you for joining again. So I'm making sure that you will follow up questionnaire to name to, and you will be able to, your recording will be available to you, and it will be emailed to you in a few days time. Plus you also have my email, so if you have any questions, you'll be able to address that too. With better space transport system, shielding and artificial gravity prevents a CVD possibility. See, the cardiovascular disease, the question is that the shielding can do part of the radiation. As I told you, there are two effects. One is the radiation and the microgravity part. So the microgravity part will be very difficult because you are at space, you will be exposed to microgravity. The radiation can be done, but keep in mind that you will be doing a lot of spacewalks because that will be a major task. So yes, shielding, but think of it, the extra shielding comes with your body weight and the logistics of the space suit. So it is sort of an optimized dice problem. Yes, it can be reduced, but I don't think so it can be eliminated. So you have to be in the top of it. So again, thank you, and look for the email for the recording in the next few days. Thank you for joining.